welcome back to what I did in lab last week, where I tell you guys what I did in my biochemistry lab. I did not do it last week because I was sick all weekend again, but this is what I did. If you remember from my last video about this lab, which was now 2 weeks ago, my lab partner and I extracted some RNA from some lavender plants, specifically the flowers. Then we extracted it, purified it, and turned it into cDNA, which worked against the odds because we accidentally messed up part of the lab, but it worked. We had viable cDNA to use in this lab. Now, this lab was determining the expression of a specific enzyme, Lin-S, to see if it's expressed in any different quantities between the leaves of the lavender plant and the flowers of the lavender plant. So we had the flowers, so that's what we were looking at. So basically what we did was take the cDNA sample that we made last week and add some primers to it that help express the specific enzyme, protein, whatever. So we had some primers that were provided to us in the lab that bind to the cDNA to measure the levels of expression of the Lin-S protein. So what we did, we had a master mix that included our cDNA, some things that were needed for the PCR reaction, and the enzyme primers for the Lin-S, as well as an actin housekeeping protein. So we used actin as a housekeeping gene to kind of normalize gene expression. The actin gene is supposed to be expressed in the same level between the leaves and the flowers, and the Lin-S is supposed to be in a different level. So the actin is just there to compare the level of expression between the two different tissue types. So once we were done creating the master mix, we all mixed it up and left it in the PCR thermocycler polymerase chain reaction to amplify and get the expression and everything all done. While we were waiting on the PCR cycles to finish, we made an agarose gel to run our gel electrophoresis on, because that's how we determine the level of gene expression. Then we just kind of waited for the gel to solidify and wait for the PCR to finish its cycles, and then we took the now primed and everything cDNA out of the thermocycler and added the loading dye, and then loaded it into the gel and let that run. There was a whole lot of waiting during this lab. We had to wait for the PCR to be done, we had to wait for the gel to solidify, we had to wait for the gel electrophoresis to run its whole thing, but by the end, so this is what we were left with. So we have a bunch of different lanes here. These two are ladders, which are used as a comparison to see how long each DNA fragment is. Now, this is actually showing two different groups. We have my group, which was lavender flower tissue, and another group, which is lavender leaf tissue. So we had two samples from my lavender flower tissue, two samples from the leaf tissue for the Lin-S, and then two samples from the lavender actin, and two samples from the lavender flower actin, and then the two samples from the lavender leaf actin. There we go. So as you can see, the actin expression is relatively the same. Ours is a little bit thicker just because we had a bit more RNA to work with initially, so we just had more stuff in the sample, so it shows up as darker and thicker. That is to be expected because it is the housekeeping gene, so it should be expressed to relatively the same rate in both the flower and the leaf tissue. However, the Lin-S is also how it's expected to be, but you can see in the leaf tissue, there is little to no expression of the Lin-S at all, whereas in the flowers, there was quite a bit of it. It has quite a strong band. So what those results mean is that actin, the gene, was expressed equally in both the leaves and the flower tissue. However, Lin-S was only expressed in the flower tissue. So what the Lin-S gene codes for is actually a linalool synthase gene, so it codes for the synthesis of linalool. So linalool is a terpene alcohol that's found in many fragrant smelling plants and spices and all that. It's part of the molecules that give lavender its scent, and it's also in a bunch of other fragrant plants, but it's part of what makes lavender smell like lavender. So the flowers are the more fragrant part of the plant, so it does make sense that it's expressed in much higher levels in the flower than it is in the leaves. So yeah, that is what I did in lab last week. There will be a new one coming out this week because I have an entire new lab starting on Friday.